Let's do a review again. Biological classification. I'll just read it fast. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done instinctively, not using criteria that were scientific but born out of need to use organisms for our own use. For food, shelter, and clothing, Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with the kingdom of Plantae and Animalia was developed that included all plants and animals. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, photosynthetic and non-photosynthetic organisms. Classification of organisms into plants and animals was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification was for a long time found inadequate. Used for a long time was found inadequate. Besides, gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationships, etc. Classification systems for the living organisms have hence undergone several changes over the time. Though plant and animal kingdoms have been a constant under all different systems, the understanding of what groups, organisms be included under these kingdoms have been changing. The number and nature of the other kingdoms have also been understood differently by different scientists over the time. The characteristics of the five kingdoms in the Monera, except Monera, all the other four kingdoms are eukaryotic, where Monera is prokaryotic. Cell wall in Monera is non-cellulosic and polysaccharide with amino acids. Protista, some had the cell wall but some didn't. Fungi, the cell wall is present and is made of chitin. It is, we can say, a chitinaceous cell wall. Plantae, it is present and it is cellulosic, meaning it is made of cellulose. Animalia is the only kingdom that doesn't have a cell wall. Nuclear membrane, except Monera, every other kingdom has a nuclear membrane. Body organization, Monera and Protista are unicellular, strictly unicellular. Fungi is multicellular, loose tissue. Plantae is tissue organ, and Animalia is tissue, organ, and organ system level of body organization. Mode of nutrition, the Monera kingdom can be both autotrophic, under which we have chemosynthetic and photosynthetic, along with heterotrophic, whereas Protista is photosynthetic, autotrophic, which comes under autotrophic, and it can also be heterotrophic. And we have fungi, which is heterotrophic, saprophytic or parasitic. Plantae, it is mostly autotrophic, and Animalia, it is heterotrophic, holozoic or saprophytic. Holozoic referring to eating or swallowing of the food. R.H. Whittaker in 1969 proposed a five-kingdom system of classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him included cell structure, body organization, mode of nutrition, reproduction, and evolutionary or phylogenetic relationships. The three-domain system has also been proposed that divides the kingdom of Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain, and thereby a sixth kingdom classification. Let us look at this five kingdom classification to understand the issues and considerations that influence the classification system. Earlier, classification systems included bacteria, blue-green algae, fungi, mosses, worms, gymnosperms, and angiosperms under plants. The character that unified this whole kingdom was that all organisms had a cell wall in their cell. This placed together groups which widely differed in other characteristics. It brought together the prokaryote bacteria and blue-green algae cyanobacteria with other groups which were eukaryotic. It also grouped together unicellular organisms and the multicellular ones, say for example chlamydomonas, which comes under cyanobacteria, and spirogyra were placed together under algae. The classification did not differentiate between heterotrophic group, which is the fungi, the autotrophic green plants, though they also showed a characteristic difference in their cell wall composition. The fungi had chitin in their walls, while the green plants had a cellulosic cell wall. When such characteristics were considered, the fungi were placed in a separate kingdom, the kingdom of fungi. All prokaryotic organisms were grouped together under the kingdom of Monera, and the unicellular eukaryotic organisms were placed in the kingdom of Protista. The prokaryotic organisms which we see can only be found in the kingdom of Monera, and as for Protista, we can find unicellular eukaryotes. All the unicellular eukaryotes are placed under the kingdom of Protista, which we call as the members of the Protista, which we call as protozoans. Kingdom Protista has brought together Chlamydomonas, chlorella, earlier placed in algae with plants, and because both were having cell walls, under Plantae it was chlorella first was placed under the kingdom of Plantae because chlorella was found to have a cell wall. Chlamydomonas, chlorella with paramecium and amoeba, which were earlier placed in the animal kingdom which lacked cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. Earlier, Chlamydomonas, chlorella were placed under Plantae, whereas paramecium and amoeba were placed under animals. But now, as they are unicellular eukaryotes, these four organisms have been placed under the kingdom of Protista and are called protozoans. This placement happened because the criteria for classification changed. This kind of changes will take place in the future too, depending on the improvement in our understanding of characteristics and evolutionary relationships. The more we understand, the more we can define a specific organism more easily, and we can classify them based on what is suited for their characteristics. Over time, an attempt has been made to evolve a classification system which reflects not only on the morphological, physiological and reproductive similarities, but is also phylogenetic, that is based on evolutionary relationships. In this chapter, we will study characteristics of kingdom Monera, Protista and fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapters 3 and 4. The first main topic being the kingdom of Monera. Bacteria are the only members of kingdom Monera. They are the most abundant microorganisms. We can say that bacteria are cosmopolitan. Anywhere you see, you will find bacteria. As such, we call it as cosmopolitan species. Bacteria occur almost everywhere. Hundreds of bacteria are present in a handful of soil. They also live in extreme habitats such as hot spring, desert, snow and deep oceans where there are very few other life forms that can survive. Many of them live in or on other organisms as parasites. Bacteria are grouped under four categories based on their shape. The, the spherical round shaped one are called coccus or cocci bacteria, where the rod shaped one are called bacillus bacteria, which we can take example for streptococcus, lactobacillus, this we can take cholera, and these are the shapes where spirilla is the spiral shaped and vibrio is the comma shaped bacteria. The spherical coccus, plural cocci, the rod shaped bacillus, plural bacilli, the comma shaped vibrium, plural vibrio, and a spiral spirillum, which is plural spirilla. Though the bacterial structure is very simple, they are very complex in behavior. Compared to this statement is to be remembered, though the bacterial structure is very simple, they are very complex in behavior. Compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. This statement to be remembered as well. Two statements that though bacteria are structurally simple, they are very complex in terms of behavior, and compared to many other organisms, bacteria as a group show the most extensive metabolic diversity. Some of the bacteria are autotrophic. Metabolic diversity meaning they can get food, like some of the bacteria are autotrophic, they synthesize their own food from inorganic substrate. They may be photosynthetic autotrophic or chemosynthetic autotrophic. The vast majority of bacteria are heterotrophs, that is they depend on other organisms or dead organic matter for food. And one of the topics which is under archaebacteria, and which is if you remember, we studied something about the three domain systems which divided the kingdom of Monera into two kingdoms, into two divisions or two domains, which is the archaebacteria and the eubacteria, where we are going to study now about the archaebacteria. Archaebacteria meaning ancient bacteria. These bacteria are special since they live in some of the most harsh habitats, such as extreme salty areas, which we call as halophiles, hot springs, thermoacidophiles, which have high temperature as well as pH conditions, extreme temperature and pH conditions, pH meaning acidity or basicity, and marshy areas, which is what we call methanogens. Archaebacteria differ from other bacteria in having a different cell wall structure, and this feature is responsible for their survival in extreme conditions. What is difference between archaebacteria and eubacteria, if you ask? The difference is that archaebacteria have a special cell wall. This special cell wall structure helps in protecting the archaebacteria and helping them survive in these extreme conditions. Methanogens are present in the gut of several ruminant animals such as cows, buffaloes, and they are responsible for production of methane biogas from dung of these animals. These animals, archaebacteria, under the category of methanogens, are found in gut of animals like cows, buffaloes, etc., and they can act as chemosynthetic bacteria and produce their own food. By doing so, they produce methane, CH3, from the food consumed by the organisms, and this can also be from the dung of the animals. This can also be seen from the dung of the animals. And the next main topic being eubacteria. There are thousands of bacteria or different types of eubacteria or true bacteria. They are characterized by the presence of a rigid cell wall and, if motile, a flagellum. The cyanobacteria, also referred to as blue-green algae, have chlorophyll A similar to green plants and are called photosynthetic autotrophs. The cyanobacteria are unicellular, colonial or filamentous, freshwater, marine or terrestrial algae. Basically, there are many hundreds of thousands of types of bacteria, and under the true bacteria or eubacteria category, it is categorized if they have a rigid cell wall and if they are motile with the presence of flagellum, and the cyanobacteria, which we refer to as blue-green algae and which were earlier placed under the plants or the kingdom of Plantae, have a chlorophyll A similar to green plants and are photosynthetic autotrophs. The cyanobacteria are unicellular. They may be unicellular, as an example for Chlamydomonas, or they may be colonial or filamentous. They are unicellular, but they form colonies where they are aggregates. They group together to form colonies, or in this case, it is filamentous. A filamentous blue-green algae nostoc. This is the mucilaginous sheath, which may be due to prevent wetting of the bacteria, and this heterosis structures, these structures in these filamentous algae is what we call heterosis, and they convert atmospheric nitrogen or fix nitrogen. The colonies are generally surrounded by gelatinous sheath. They often form blooms in polluted water bodies. Some of these organisms can fix atmospheric nitrogen in specialized cells called heterosis, example nostoc and anabina. Chemosynthetic autotrophic bacteria oxidize various inorganic substances such as nitrates, nitrites, and ammonia, use the released energy for their ATP production. They play a great role in recycling nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria. That paragraph is for autotrophic bacteria. The first part being photosynthetic, and the second part being chemosynthetic, where the chemosynthetic bacteria get energy from oxidizing various inorganic substances such as nitrates, nitrites, ammonia, and the energy released from this oxidation is what they use for their ATP production. They recycle nutrients like nitrogen, phosphorus, iron, and sulfur. Heterotrophic bacteria are most abundant in nature. The majority are important decomposers, many of them saprophytes actually, have a significant impact on human affairs. They are helpful in making curd from milk, production of antibodies, fixing nitrogen in legume plants, the nodules, roots, etc. Some are pathogens causing damage to human beings, crops, farms, animals, and pets. Chlorella, typhoid, tetanus, citrus canker are well-known diseases caused by different plants, where chlorella, typhoid, and tetanus are human, and citrus canker being a plant disease. Bacteria reproduce mainly by fission. This is a dividing bacteria. This is the cell membrane. This is the cell wall. This is the nucleoid. and this is the junction. Reproduce mainly by fission. Sometimes under favorable conditions they produce spores. They also reproduce by a sort of sexual reproduction by adopting a primitive type of DNA transfer from one bacteria to other. The mycoplasma are organisms. The next topic which comes under the eubacteria, or the last topic that comes under the eubacteria in this textbook, or referring to NCERT at least, is the mycoplasma. Mycoplasma are organisms that completely lack a cell wall. They completely lack a cell wall. They are the smallest living cells known and can survive without oxygen. Many mycoplasma are pathogenic in animals and plants. 2 points to be remembered: lack cell wall, can survive without oxygen. Of course, the third point is smallest living cells. And a fourth point you might or you should remember is pathogenic in animals and plants, at least most of them. And I believe the size was 0.3 to 5 micrometers, but I'll see. Kingdom Protista. All single-celled eukaryotes are placed under Protista, but the boundaries of this kingdom are not well defined. What may be a photosynthetic protist to one biologist may be a plant to another. In this book, we include chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans under the Protista. Members of Protista are primarily aquatic. This kingdom forms a link with others dealing with plants, animals, and of course with fungi. Being eukaryotes, the Protistan cell body contains a well-defined nucleus and other membrane-bound organelles. Some have flagella or cilia for motility. Protists reproduce asexually and sexually by a process involving cell fusion and zygote formation. This is a sort of sexual reproduction we can even see in kingdom of Monera, where there is a primitive method of DNA transfer. It might be the same here or similar. Chrysophytes. This group includes diatoms and golden algae. Desmids. They are found in freshwater as well as in marine environments. They are microscopic and float passively in water currents, which we call as a plankton. They can be phytoplanktons, meaning plant-related planktons, or plankton, meaning particles which are living. Microscopic organisms which float passively in water current is what we call a plankton. Zooplanktons come under the kingdom of no, they don't come under animalia. Most of them are photosynthetic. In diatoms, the cell wall form two thin overlapping shells which fit together as in a soap box. The walls are embedded with silica, and thus the walls are indestructible. Thus diatoms have left behind large amount of cell wall deposits in their habitat. This accumulation over billions of years is referred to as diatomaceous earth. Being gritty, this soil is used in polishing, filtration of oils and syrups. Diatoms are the chief producers in the ocean. So the group chrysophytes includes diatom, golden algae, which we call as desmids. They are found in both freshwater and marine water environments. They are microscopic and float passively in water. Most of them are photosynthetic. In diatoms, the cell wall is actually similar to a soap box with two shells overlapping, and these shells are embedded with silica, and with this embedment of silica, the walls become indestructible. As the desmid continues to die and it continues to float drown into the ocean bed or the seabed, gets continuously, these cell walls get continuously accumulated, and this accumulation is what we call a diatomaceous earth. For billions of years, this accumulation is what we call diatomaceous earth. This diatomaceous earth being gritty, the soil is used in polishing, filtration of oils and syrups. Diatoms for producing this diatomaceous earth are called the chief producers in the ocean. And the shell is actually called frustal. The two overlapping shells is what we call a frustal. The next topic under Protista or kingdom Protista is the dinoflagellates. These organisms are mostly marine and photosynthetic. They appear yellow, green, brown, blue, and red depending on the main pigments present in their cells. The cell wall has stiff cellulosic plates on the outer surface. Most of them have two flagella. One lies longitudinally and other lies transversely in a furrow between the wall plates. Very often red dinoflagellates, example goniolax, undergo such rapid multiplication that they make the sea appear red. This is what we call red tides. Toxin released by such large numbers may even kill other marine animals such as fishes. The dinoflagellates are basically marine and photosynthetic organisms. They appear in many colors depending on the pigment present in their cells, and they have two flagella, one which is horizontal or latitudinal and other which is a longitudinal flagella, which is in a furrow or a groove which runs longitudinally between the wall plates. These wall plates or stiff cellulose plates, what we call, are made of cellulose. And another information given here is that red dinoflagellates, when they undergo multiplication, they undergo such rapid multiplication that the toxins they release during this multiplication make the sea appear red in color, along with kill many fishes or other marine animals. The next topic is euglenoids and the protozoans. Protista, majority of them are freshwater organisms found in stagnant water. They are freshwater organisms but are found not in flowing water but stagnant water. Instead of a cell wall, they have a protein-rich layer called pellicle, which makes their body flexible. They have two flagella, a short and a long one. Though they are photosynthetic in the presence of sunlight, when they are deprived of sunlight, they behave like heterotrophs by predating on other smaller organisms. Interestingly, the pigments on euglenids are identical those present in higher plants. Example is euglena. Euglenoids, example euglena, are freshwater organisms but present only mostly in stagnant water. They have a protein-rich layer called pellicle instead of a cell wall, and this pellicle layer is very flexible, and they have two flagella of one long and one short, and they behave as both autotrophic and heterotrophic organisms. Autotrophic, they have pigments similar to higher plants and can do photosynthesis in the presence of sunlight, and if deprived of sunlight, they predate on other organisms. This is heterotrophic behavior. Next topic is slime molds. Slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic matter. Under suitable condition, they form an aggregation called plasmodium which may grow and spread over several feet. During unfavorable conditions, the plasmodium differentiates and forms fruiting bodies bearing spores at their tips. The spores possess true walls. They are extremely resistant and survive for many years. Even under adverse conditions, the spores are dispersed by air currents. These slime molds are saprophytic protists. The body moves along decaying twigs and leaves, engulfing organic matter, which means that they depend on dead and decaying matter. They, under suitable condition, they form aggregation called plasmodium, which may grow and spread over several feet. This is the aggregation or grouping of the several slime mold, which happens only under favorable conditions. This aggregation or spreading of over several feet is what we call plasmodium, and during unfavorable condition, this plasmodium which has spread will differentiate into fruiting bodies and differentiates and form fruiting bodies bearing spores at their tips. These spores possess true cell walls. They are extremely resistant and survive for many years, and they are dispersed by air currents. Protozoans. All protozoans are heterotrophs and live as predators or parasites. They are believed to be primitive relatives of animals. There are four major groups of protozoans. Amoeboid protozoans, which we can take as amoeba as an example. These organisms live in freshwater, sea, or moist soil. They move and capture their prey by putting out pseudopodia, false feet, as in amoeba. Marine forms have silica shells on their surface. As we saw in the chrysophytes, these organisms also have, the marine forms have silica shells. Some of them, such as ant amoeba, are parasites. Ant amoeba. Flagellated protozoans. The members of this group are either free-living or parasitic. They have flagella. The parasitic forms cause diseases such as sleeping sickness. Example is trypanosoma, which is a flagellated protozoan, which is a point to be remembered. Ciliated protozoans, which is another category under the protozoans. These are aquatic, actively moving organisms because of the presence of thousands of cilia. These are aquatic organisms, actively moving around big organisms, big presence of thousands of cilia. They have a cavity called gullet that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water laden with food to be steered into the gullet. Example is paramecium. This furrow here, or a hole or a cavity, is what we call a gullet, and cilia the paramecium will capture its prey, and the food will be, with the coordinated movement of cilia, will be pushed inside the gullet. These are what we call contractile vacuoles. There are two nucleus of the macro and the micro nucleus. These are contractile vacuoles. This is the place for excretion. As for flagellated protozoans, the main thing to be remembered is they can either be parasitic or free living, and the parasitic version is that they are disease caused, which is sleeping sickness, which is caused by trypanosoma, and this is to be remembered. Trypanosoma, flagellated protozoans. And ciliated protozoans, they are aquatic, actively moving. We can take example for paramecium. The last type of protozoan, this sporozoans. This include diverse organisms that have an infectious spore-like stage in their life cycle. The most notorious is Plasmodium, malarial parasite, which causes malaria, a disease which has a staggering effect on human population, which has an infectious spore-like stage, is what we call sporozoans. And this is to be remembered that under the sporozoans comes Plasmodium, which causes the malaria disease. And the third main topic under the biological classification is the kingdom of fungi. The fungi constitute a unique kingdom of heterotrophic organisms. They show a great diversity in morphology and habitat. You must have seen fungi on a moist bread, rotten fruits. The common mushroom you eat and toadstools are also fungi. White spots seen on mustard leaves are due to parasitic fungi. Some unicellular fungi, example yeast, are used to make bread and beer, alcohol. Other fungi cause diseases in plants and animals. Wheat rust causing Puccinia is an important example. Some are the source of antibiotics, example penicillium. Fungi are cosmopolitan and occur in air, water, soil, and on animals and plants. They prefer to grow in warm and humid places. Have you ever wondered why we keep food in a refrigerator? Yes, it is to prevent food from going bad due to bacterial or fungal infections. With the exception of bees, which are unicellular, fungi are filamentous. With exception of yeast, we saw that the fungi body organization is loose tissue organization or tissue organization, which means it is multicellular, but yeast is the only one that is unicellular. Fungi are filamentous, like thread-like structures. Their bodies are consistent of long, slender, thread-like structures called hyphae. The network of hyphae is what we call or is known as mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. The hyphae can be multinucleated, meaning there are no divisions, and this goes on. If this is a hyphae, there will be nucleus here and there will be no separate divisions. So we can call it as aseptate or multinucleate, or in other words, cenocytic hyphae or mycelium. Some hyphae are continuous tubes filled with multinucleated cytoplasm. These are called cenocytic hyphae. Others have septae or cross walls in their hyphae. Here, this is cenocytic. No septae or separation, whereas in another case, if this is a hyphae is a kind of we can assume it to be a root-like structure, or which is a part of cells. This is what we call a septae, and here it is not multinucleate, but each cell has only one nucleus, or it is specific to the individual organism, but septae is there. The cell walls of fungi are composed of chitin and polysaccharides. Most fungi are heterotrophic and absorb soluble organic matter from dead, and hence are called saprophytes. And those that depend on a host, which are plants, living plants, and animals, are called parasites. They can also live as symbionts, mutual relationship in association with algae as a microbiont and a phycobiont, which together form with a mutual relation as a lichen, which you have as a last topic, and with roots of higher plants as mycorrhiza. Reproduction in fungi and takes place by vegetative means. Vegetative means meaning if we cut a plant or a vegetative part and plant it somewhere, or like if we cut the stem of a plant and plant it somewhere else, a new plant will arise from the stem of the other plant. This is what we call a vegetative means of asexual reproduction or fragmentation, as we see in Spirogyra, where the Spirogyra, if it fully matures, it will get fragmented into numerous new Spirogyra, along with fission, division, as we see in Monera, kingdom Monera, where bacteria divide, budding, as we see in Hydra, where a bud develops, asexual reproduction by spores. These are the whatever we saw: fragmentation, fission, and budding. These come under the vegetative means. As for the asexual reproduction is by spore formation, called conidia or sporangiospores or zoospores. And sexual reproduction is by oospores, ascospores, and basidiospores. Conidia or sporangiospores or zoospores come under asexual reproduction, whereas oospores, ascospores, and basidiospores, these come under sexual reproduction. The various spores are produced in distinct structure called fruiting bodies. What produces spores? Spores are produced by fruiting bodies. Fruiting bodies produce spores. Fruiting bodies produce spores. The sexual reproduction or sexual cycle involves the following three steps: fusion of protoplasms between two motile or non-motile gametes. The sexual cycle involves three steps. How sexual cycle happens or sexual reproduction happens is, the first step is fusion of protoplasm. Protoplasm is the cytoplasm along with the nucleus, which is what together we call nucleus plus cytoplasm as the protoplasm. Fusion of protoplasms between two motile or non-motile gametes called plasmogamy. Fusion of two nuclei called karyogamy. Meiosis in zygote resulting in haploid spores. These are the three steps, where first step being fusion of protoplasm, which is what we call as plasmogamy, and fusion of two haploid gametes or two nuclei, which we call it as karyogamy, and meiosis in zygote resulting in haploid spores. An example of fungi reproducing sexually is when a fungus reproduces sexually, two haploid hyphae of compatible mating types come together and fuse. If these are two hyphae are network-like structures, and together a network of hyphae is what we call mycelium. If these are two hyphae and they are haploid, N, this is an N, meaning they are haploid. If they are compatible, condition being compatible, haploid, then these are two myceliums of two different fungi of compatible type, then they fuse to form a completely new hyphae. This is a means of sexual reproduction. A means of sexual reproduction in fungi. This condition being they should be of the compatible type, they should be haploid, and two mycelia two hyphae should fuse. In some fungi, the fusion of two haploid cells immediately results in in a diploid cell 2n. However, in other fungi, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occurs. Such a condition is called dikaryon, and the phase is called dikaryophase of fungus. Normally, when there are two cells of n ploidy haploid and they fuse, they form a 2n diploid cell. But in certain cases of under the fungi, there are categories under which there come Ascomycetes and Basidiomycetes. For these fungi, if two haploid cells, let's assume this is the nucleus, this is the nucleus, and they fuse, they are both n and ploidy, and they fuse, they don't, the nucleus, the karyogamy will not happen immediately, and there will be two nucleus in the cell. This ploidy we represent as n plus n, and this stage is called the dikaryophase. And yes, Ascomycetes and Basidiomycetes, an intervening dikaryotic stage n plus n, that is 2 nuclei per cell, occurs. Such a fusion or condition is called dikaryon, and the phase is called dikaryophase of fungus. Later, the parental nuclei fuse and a cell become diploid. The fungi form fruiting bodies in which reduction occurs, leading to formation of haploid spores. Later, after this stage, this cell will again fuse, the nuclei will fuse to form a 2N cell or a diploid cell. This will divide mitotically to produce haploid spores. Meiotically to provide haploid spores. The morphology of the mycelium, mode of spore formation and fruiting bodies form the basis for the division of this kingdom into various classes. The three main categories for dividing the kingdom of fungi into into various categories is mode of spore formation, morphology. As we saw, the mycelium, which is the network-like structure of hyphae, can be the cenocytic multinucleate or septate, which will have uninucleate. So one is the structure or morphology of mycelium, mode of spore formation, how the spore is formed. As we saw in asexual reproduction, it is followed by sporangiospores, zoospores, or asexual spores called conidia, sporangiospores or zoospores. And sexual reproduction in fungi is by oospores, ascospores, or basidiospores. And these various kinds of spores are produced in distinct structures called fruiting bodies. In ascospores, in ascomycetes, a special structure called asci, and basidio, it is basidiocarp. And these are the few images they gave of A. It is mucor, of B. aspergillus, and C. This is the basic mushroom.